Hello everyone, welcome back to the Biology 3157 Lecture 3 is about landscape genomics. I'm going to use some examples today from a project that we've been working on in eucalyptus. This is a collaboration between several groups, Linda Broadhurst at CSIRO, my lab, and also there's a Western Australian team, which we're not going to talk about today. It's looking at the foundation species on the east and west coast. This is yellow box, Eucalyptus meliodora, and the samples come from a set of citizen scientists. These are often beekeepers at all those yellow spots that have sent in leaves and sometimes seeds into the Australian Tree Seed Center. And those leaf samples came across to our lab. We prepared the DNA and sequenced them using genotyping by sequencing to identify the genetic diversity. And we had about 37 sites, 300 trees, and more than 10,000 SNPs. So one of the first things we did now was this principal component analysis that you guys have just done as well. And we colored the samples by the site at which they came from. And so on the left, you can see there was a site that comes from near the Queensland border, so it's northern site, and then three more sites that go from basically Canberra out to Wagga Wagga, east-west gradient. And when you put these trees into an unsupervised principal component analysis, the genotypes by samples, they seem to split out pretty nicely by sites, both the north-south and an east-west gradient. They're in the second principal components. So we're seeing some modest population structure where samples are clustering by sites. And on the right, we have a within family analysis. So there was one particular tree that was of interest to folks because it had it was a mosaic tree actually. It had one branch that was completely defoliated by Christmas beetles, whereas the rest of the tree had plenty of leaves. And so we're wondering what was wrong with that branch. Could it have been a mutation or an epigenetic change? So we collected seeds off of that branch as well as leaves off of that mother tree and other potential fathers in that site, in that plot. Leaves were also collected. So we put those through, 96 samples or so, through the genotyping by sequencing platform, and then did a correlation on the samples, both the parental samples, the fathers and the mothers, against the seedlings, the progeny that came off. And so in the rows are the seedlings, and in the columns are the parents. And anytime there's a yellow box, it's when there's a high correlation between the parent and the offspring. So this is a paternity test. You can see that that first column, the father had three children, and the second father might be slightly more related to the mother because there's a bit of an orange kind of higher background level, but he certainly fathered one. The next father was probably even closely related to the mother, and it's kind of a little less clear in that case. And then you got two seeds and 1 and 0 and 0 and 1. That's the parentage. So not all the fathers contributed, some fathers more than others, and even the mother was the father in a few cases. These white stripes, it's just 4 leaves off the same mother plant. The white stripes are the self-pollinated seeds where the father was the mother. So we can see that there's a little bit of inbreeding, some outcrossing, usually with fathers that are nearby, but often father was not found. So long-range pollen flow, and not all fathers might have been flowering at the same time, so they didn't necessarily contribute. So this is a macro look at 4 populations across the range, and a micro look at the genetic variation within a family. Now, that's the simple story. It's going to get more complicated, as it always does. So we wanted to get more sites and span the complete range and not have these big north, south, east, west gaps, but expand the collection. And we also acknowledge that there's quite a bit of morphological diversity if you just look at the leaf shape from these different places. this is a dendrogram. Now the outgroup, you can see at the top, just like what we did in lab with the brachypodium samples, there's a population O and one sample M that is completely distantly related, and these samples from site 5, site 26, they are maybe hybrids, but they're not this pure species that we were interested in. And then you get down to all this collection. At the bottom, we've got A711. That's 2 trees, which really probably were the same tree in site A. Here's 19, 8 and 9. And then over here we can see site 7, plant 4, and its replicate. So this is what a known technical replicate looks like. 2 leaves from the same tree. And here's 2 trees, which we thought were different, tree 3 and tree 5 from site 11. They actually were the same tree, probably connected by the roots. So there's quite a few examples that we found where trees really were not independent trees, and some sites and trees that were not even Eucalyptus meliodora. So we wanted to filter through both those clone groups and remove the outliers, and they come up with a filtered population. And this filtered population we can look at now on a principal components plot, and the axes are explaining very little amounts of variation, 1.3, 1.7%. There's huge amounts of diversity in here that cannot be simply summarized by a north, south, east, west kind of projection of genetic space. These are all the individual trees, but they're color-coded by the rainbow colors from north to south, so red being north and blue, purple being south. And you can see there's a bit of geographic signal, even though there's not any clear distinct boundaries as in the previous example. Now, one thing that's often done is to ignore the within site variation, and most people just collapse all the individuals within a site and compare the genetic distance between sites. This is not between individuals now, this is just between sites. And you do this with a statistic called FST, which is the allele frequency difference between sites. So the SNPs now are averaged to get a number between 0 and 1, which is the amount of variation that can be explained by sites and allele frequency at every single SNP. And that summary statistic now also shows little geographic signal, but it can be summarized 11% or 25% by these first 2 principal components. So that's a complicated distinction. Basically, if you want to treat and acknowledge the individual levels of variation, then there's not a lot of differentiation between sites. But if you focus now on just the differentiation between sites, then you can start to explain more of that variation. So that is a little bit confusing, but it's just I want to bring that up because most people are not doing individual genotyping, and now with the basically new methods for genomic analysis, we can start to appreciate all of that huge amount of genetic diversity within sites and relax the assumption of the fixed differences between sites as the major signal. Acknowledge all the variation that exists in the local site. Now, once you have this matrix of differentiation among sites, we're going to go now from the landscape genomic analysis. There's 2 parts to the landscape. There's the geography, which is just the distance in kilometers down at the bottom, and then there's environment. So isolation by distance is kind of a neutral process, where seeds, apples don't fall far from the tree. This is eucalyptus seeds. The seeds don't travel too far, but in this case, the pollen's traveling much farther than we thought. So 500 kilometers is the geographic range upon which we start to notice a little bit of difference. And this is not a huge amount, but out at 1,000 or more kilometers, this is the extremes between the far north and the far south of our sampling sites. We start to see genetic differentiation out at that huge range. So this is really changing the paradigm completely of local adaptation. And most people are focusing on collecting their eucalyptus seeds either within their site or within 50 kilometers or something to maintain the locally adapted genotypes. But these populations are so big, and this is just the signal between sites that we're trying to explain. There's huge amounts of gene flow, and so whether it's swift parrots or other processes, but there's gene flow across this wide range, and they don't all flower at the same time, and they live for a long time. So these trees really are part of large populations, and we may be artificially restricting gene flow by sampling from such small locations. Now, just to show like what are the um, this fit, would it occur by chance? What if we did a jackknife or a bootstrap and left some sites out? How steep is this slope? Is it just um, the result of the particular sites that we chose? What if we were to resample the actual sites and refit the curves? And so this is 90 or 100 permutations, resampling a subset of the sites and fitting the curve. And so you can see the confidence in that isolation by distance in panel G and H here. Now above, we're looking at environmental units. There's four particular variables that can be extracted from the GPS coordinates. So the amount of rain at each site, precipitation in the wettest quarter, isothermality, which is the day-night temperature difference divided by the winter-summer temperature difference. So Canberra basically is got high isothermality because the summer winter is relatively small compared to the diurnal shift. That's important, but not till you get to high values of isothermality. There's temperature in the coldest quarter had an important effect on genomic distance between, say, 6 and 10 degrees. Nitrogen in the soil at about 30 centimeters below ground was also important. So we got a mixture of climate signals, precipitation, temperature, and seasonality, as well as soil, nitrogen in this case. Those environmental units, along with geography, together can do a pretty good job of explaining the observed genomic distance between these samples. And we can flip this around now. We have this generalized dissimilarity model that includes both geographic and environment, and you guys are going to use a simpler version called the Mantell test, the partial Mantell test that includes the geographic distance in kilometers, pairwise distance between sites, and an environmental matrix, which can include up to 20 commonly used bioclimatic variables. Not all of them will be important. So together, isolation by environment and isolation by distance can explain the observed genetics. And that's then we're learning about the factors that control the relatedness among these populations. This is landscape genomics. So when we invert the model now, we'd like to use our high-resolution maps of environmental variables and the geographic distance between all these pixels and project the genetic distance. This helps us interpolate where the ranges might be. This is a massive interpolation from 37 sites out to billions of pixels here in beautiful colors. So we're using 3-color space to kind of represent the genomic distance in red, blue, and green. And you can sort of recognize the environments of the alpine region has got a different expected genomic signature to the arid inlands and the more tropical north coastal regions of Australia. So isn't that beautiful? This is why you'd like to fit a model of genomic distance by geography and environment, so then you can make projections of what the genomic distance might be in areas where you've never seen it. And if you do those projections in the current climate, as well as in the future climate, so we have climate models that can tell us what we think the temperature and rainfall and isothermality will be at every pixel. should we project our genomic distance into the future environment as well as the present environment, and then overlay the two and look at the distance? Where will be the fit of current genetic variation and future genetic variation? That's what we're looking at here. So the green spots are where there's going to be a lot of change. And the green spots there is where there's a big difference between the current one, which is on the left, and this future one in 2770. And so those sort of green spots are kind of mid-latitudes where the summer rainfall zone is moving south. Essentially, the Mediterranean climate of Victoria is getting pushed off the map in the future. And although the Alpines are probably still a good refugia, there's very little difference in the projected genetic distance between the future and the present in the Alpine regions, but very large genetic differences predicted for the future. So that basically means we should change our seed sourcing to try to find habitats that will be suitable in the future. So this is assisted migration. We want to source seeds from populations and plant them in areas where the climate will be suitable in the future. And this is what, if you're sourcing for a restoration site in this black dot here in the south on the left, you project that anywhere in green would be a pretty good match. So that means local wouldn't be bad in that southern range. But on the right, the preferred spots would not only be in the site of restoration, but would also include some coastal sites. So this is a tool now to say, I know the pattern of geographic and environmental variation. I use that to project genetic variation from my model, and I can do that for every site in future climates. And so if I'm going to plant trees at this site, I'd like to look up where would be good places to collect seeds. So this is a pretty exciting new study that has, one, changed our view of what local is, and it's much larger than we thought before. We also know there's much more genetic diversity within the site than we thought before. And we've got a very empirical, although highly overfitted and projected map about where we should source seeds. So it's a scientific approach to seed sourcing rather than a kind of anecdotal way of let's just local is best, let's get the seeds from our same farm if we can. That's kind of a simplistic view. So what's happening in practice is that we're trying to grow up seeds from populations that across the range in a common garden, and then when they start to cross, they'll be able to interbreed and release genetic variation, new genotypes that may not have ever existed in the field before, that would have high adaptive potential and low adaptive potential. But the seed pools, this would be a broad genetic diversity of seed pool that could survive in potentially no analog environments that have never been seen before. And so this is a long-term study to try to bank existing genetic diversity and then shuffle it up for the future, for new environments. And one of these places is our own National Arboretum. I hope you've been over there. This is just overlooking the lake. If you go down by the zoo, across the highway there, you'll see there's 100 forests for 100 years, and 10 of them are the ANU research forests. They've got both spotted gum and ironbark. And we wished they'd done a little more empirical work and to choose the trees, but they were sort of in a rush to get it done for the Canberra's centenary, and they just grabbed everything they could get from the nurseries and planted these spotted gum and ironbark forests. But it's given us a chance to go back after and do the genetics as well as some phenotyping. And the phenotyping I'll just show you briefly here. This is just how tall the trees are. This one isn't a eucalyptus forest, but it's going to change at different times of the year. So you can see the fall color change for this deciduous forest as an example of a phenological trait, which means the rhythmic nature of biological events as it pertains to climate. Phenology is a when trait. When did the leaves turn? So we've got the exact hour, if you will, of the leaf changing for almost all the trees in the whole arboretum. This is a GigaVision camera that my postdoc Tim Brown has set up overlooking Dairy Farmers Hill, and it scans every hour the whole arboretum at very high resolution so that you can scan right back down to the leaf level. We also have weather sensors in there, and the trees draw water out of the water table, and the spikes then are rain events, and the seasons. We've got a few years of data now of exactly water, moisture, temperature, air temperature, humidity, and light coming down to an array of sensors, about 20 sensors scattered throughout the forests. So we've got a lot of phenotype and environmental data streaming in, and we're just adding the sequence of the tree genomes. If anybody's interested in this project, come talk to me. Genotype, phenotype, and environments. This is the matrix that we'd like to build now from Common Garden site, as opposed to, this is a human designed, balanced, empirical experiment, as opposed to the landscape genomics experiments, which are nature's experiments. Looking backward, this is a future experiment, watching the trees grow. We can take those measures of tree height and width and color and use those values to paint onto a computer model, just grabbing the one eucalyptus tree that was available in the CAD software, and this is an Unreal gaming engine, so we can virtually fly through the arboretum and see the trees, and you can come by the lab sometime and borrow the virtual reality glasses if you want to take a look. These demos are also on Tim's YouTube site. This is how you look down in through the GigaVision camera and Google Earth, and you can jump right in and zoom right down to the individual tree level, and then Tim flies his drones through the arboretum, and they take hundreds of overlapping pictures, and that's how they can create a point cloud to triangulate the height and width. And it's almost a one leaf level point cloud, and this is open source academic tools for mapping the arboretum in 3D. Ultimately, we get a value for each tree. We want to give this lecture in class. Here's the virtual reality view of it. It's a beautiful little walkthrough. You can jump down on the ground and see the actual trees, and then we can put the environmental layers on as well. So this is just a way of visualizing very rich data sets and get kind of a feel for it. Ultimately, we're even thinking about augmented reality, where you could put on the glasses, the see-through glasses, and walk right into the forest and get all this digital information, soil information, be able to look literally into the ground, air humidity channels, as you're walking through the forest. So it's a wonderful combination of high-tech and traditional fieldwork happening right here in our backyard. It's going to be a long-term experiment. I'd love to plant another one of these arboretums, and we're working on another infrastructure proposal to do what I call a square kilometer decade. It's 100 hectares for tree planting, and we'd like to get some of the tricks that the eucalyptus people are using in the plantations, but use a diverse set of lines. So there may be more trees living off Australia than on Australia because of these high density plantations. There's certainly way more genetic diversity on Australia that hasn't yet been exported, but we're proposing to tap into that digitally with a large sequencing project. So this is also underway with collaborators from South Africa. The Australian Tree Seed Center has already tens of thousands of accessions. Some of the most widespread, Calmadulensis, river red gum, and some of the other cultivated types that are used in plantations. This is where they come from. And if you go down to the ANU coastal campus at Kyola, you can see old Splotchy here, who's probably 80 meters tall, and it takes 11 of us to put our arms around it. And this tree is probably about 500 years old, so kind of young maybe compared to some of the old redwoods in California, but one day they'll pass up those redwoods and be the tallest tree, certainly the tallest flowering plant, and amazing store of carbon, and probably below ground a similar amount. And that's really kind of the next frontier, is to look at carbon below ground and try to indirectly look at root growth if we can. So in conclusion, landscape genomics is a way of looking back in time at Mother Nature's experiments. How has genetic diversity been filtered across the landscape? And that can guide us for future restorations into new climates and new soil types. And we'd like to really scale this up at an industrial level and learn from Mother Nature about how to plant the right seeds in the right environments for building habitat, for improving soils. And that's what foundation species do, is they build the ecosystems that diversity thrives in. So, thanks for your attention.